My name is Megan Sullivan, and I'm here today to present to you our study, Effectiveness of Sacred Forests and Biodiversity Conservation, a meta-analysis. Sacred forests are forest fragments that offer co-benefits of protecting both cultural traditions and forested areas. However, sacred forests are often undervalued for their contributions to biodiversity conservation. Several qualitative reviews discuss biodiversity conservation in sacred forests, but large-scale studies that quantify the effect of sacred forests on biodiversity conservation are scarce. Many studies on sacred forest biodiversity are limited to single location censuses or lack comparisons between sacred and non-sacred forest areas that serve as controls. To fill this knowledge gap, we conducted a global meta-analysis that compared sacred forests with nearby non-sacred forest control areas and asked the following questions. 1. Will sacred forests have similar, lower, or higher levels of biodiversity as non-sacred forests? 2. Will sacred forests be more effective at protecting biodiversity than both protected and unprotected non-sacred forest control sites? And 3, will biodiversity of plant and non-plant taxa be similarly protected in sacred forests? After reviewing a total of 631 studies, we found 30 studies from 17 different countries, which both quantified biodiversity and made comparisons between sacred forests and non-sacred forest sites. Here you can see the summary of the countries that studies were from and the taxa that were studied in our meta-analysis dataset. To answer our first question, we conducted the meta-analysis on our study dataset. The x-axis on this figure shows the standardized mean difference of each study. Standardized mean difference measures the effect size. So in this case, if sacred forests have higher or lower biodiversity than control areas, found by each study in the meta-analysis. The zero line highlighted in red on this figure represents a measure of no difference. Biodiversity levels were higher when lying to the right of the zero line and lower when lying to the left of the zero line. In our study, most of the effect sizes were either on or very close to the zero line. Thus, we found that overall levels of biodiversity were similar between sacred forests and nearby forested control areas. To answer question 2 about how the protected status of the non-sacred forest control areas affected the results of the meta-analysis, we found that levels of biodiversity in sacred groves were similar regardless of the protection status of the control areas. To answer question 3, looking at how taxonomic groups affected the meta-analysis results, we found that the effect that sacred groves have on biodiversity is stronger in plant taxa than in non-plant taxa, though neither effect was significantly different than 0 in this analysis. Our meta-analysis provides quantitative evidence that sacred forests can be effective areas of biodiversity conservation. Based on our results in future studies, research on sacred forest biodiversity could compare sacred forests with other types of land use, collect standardized metadata from sacred forests and comparison sites, and extend monitoring to include more non-plant taxa. Though further data collection could serve the scientific community, we stress that not all sacred forests should be data points in a meta-analysis. Respecting traditional knowledge and the sovereignty of indigenous and local communities is paramount. Finally, sacred forests play an important role. They can preserve ecosystem function, provide social benefits, contribute to fighting climate change, and thus should not be overlooked. Thank you.